Look at these small stretches of nucleotides where one nucleotide sequence is trying to find a match or mismatch for itself by comparing with the test or the reference sequence. In simple terms, this process is called as sequence alignment. Sequence alignment is nothing else but a comparison of sequences. Welcome back to Bioenigma Uncovered. Today we are going to discuss about various types of sequence alignment algorithms, and specifically we will be focusing on dot matrix algorithm for practical purposes. So without any further delay, let's start today's video. Sequence alignment is based upon certain kind of computer programs or computer instructions which are technically called as algorithms. An algorithm is a set of instructions which is used to provide solutions to a complex problems in a stepwise manner. As far as sequence alignment is concerned, an algorithm is a bioinformatic tool which is used to compare two or more than two stretches of DNA molecules, RNA molecules or protein sequences to identify regions of similarities or dissimilarities across them. This sequence alignment algorithm will eventually suggest structural or functional or evolutionary relationships across those sequences. How do these algorithms or sequence alignment algorithms works? These algorithms works by identifying matches or mismatches or gaps between the two sequences which are being compared, that helps us in finding the best possible alignment. Sequence alignment can be categorized into various types based upon two strategies. In the first category, the sequence alignment is differentiated into two types based upon the strategy which is used for their alignment. Herein, the two types of sequence algorithms or sequence alignment are global alignment and the local alignment. Global alignment, as the name indicates, is carried out for long stretches of nucleotide sequences or amino acid sequences. This type of alignment is based upon Needleman-Wunsch algorithm, and it is carried out for those sequences which are evolutionary or phylogenetically related to each other. In this type of alignment, the sequences undergoing comparison or undergoing alignment are of identically similar length. For example, if we want to compare sequences from humans and chimpanzees, then this type of alignment would be called as global alignment. In comparison, the local alignment is carried out for those sequences which are evolutionary far apart from each other. Like say for example, we are comparing a protein sequence from humans with that of an insect. Since both these species are evolutionary not related to each other, or rather evolutionary far apart from each other, so herein we are going to focus on small stretches of nucleotides. So this local alignment is generally performed for identifying conserved domains among nucleic acid sequences or protein sequences. The other categorization of sequence alignment is based upon the number of sequences which are being aligned or which are being compared. So herein there are two categories. The first category of alignment is pairwise alignment, and the second category is that of multiple sequence alignment. We'll first talk about what is pairwise sequence alignment. As the name indicates, herein we are going to compare two stretches of nucleic acids or protein sequences. This pairwise sequence alignment can be further categorized into three types. The first is dot matrix algorithm or dot matrix method, which is a primitive one, and the second one is dynamic programming method. This dynamic programming method can be further subdivided into two types of algorithms, that is the Needleman-Wunsch algorithm and the Smith-Waterman algorithm. In the next section of this video, we are going to focus on dot matrix method. As you can see on your screen here, the two sequences which are to be compared, one of them is basically taken on a vertical axis or along the y-axis, and the other sequence which is to be compared with that is taken on the horizontal side of a grid. We are trying to align pair of nucleotide sequences which are shown as G, C, G, A, T, C, G, C, T and A, and the other sequence is A, T, C, G, C, T, A, G, C. So we will basically comparing these sequences by aligning each nucleotide when we move from along the horizontal axis on one by one. We first match A with A and we will move to the vertical side. Whenever we find the matching nucleotide, we will put a dot here. So that is why this curve is called as a dot plot. You can clearly see we get a match between A to A. Whenever the two sequences or the two nucleotides do not match with each other, we will just leave the space blank in the grid. Again, as we move towards the upward direction, we again find another A nucleotide or letter A in the vertical y-axis. Again, we will put the dot here when A matches with A. We try this method or we repeat this process for the string of letters which are lying along the positive x-axis. We'll just repeat the sequence, and within a few seconds, rather within a few minutes, you will be able to see that a nice grid is observed along the origin. This black line here indicates that there is a complete alignment between these stretch of the nucleotides. However, along this long, if you see this diagram very closely, you will see that in addition to this large or central black line, there are some additional smaller lines also present. They indicate that in addition to the global alignment, there are certain sections of sequences which are also showing local alignment. These might also indicate a repeated segment within a sequence. So overall, dot matrix method provides a really intuitive way to see the entire similarity landscape at a glance. However, dot matrix method suffers from certain limitations or shortcomings. For instance, but dot matrix plots have a big noisy problem. When you are comparing long sequences, specifically DNAs with just 4 letters, random matches do happen all the time. So a dot plot of two big DNA sequences can look like a total mess, so making it almost impossible to spot the real signal through the all background chatter. To fix this, it is suggested that a filtering method must be applied when we are analyzing two stretches of DNA molecules through the dot matrix method. So herein, instead of comparing single letters, we use a window of a specific length of nucleotides that must be employed to compare short chunks of nucleic acids. For example, you might slide a window of 10 letters from the first sequence along the second one, and then you count the matches. So you only place a dot if the number of matches is above a certain threshold. So by playing with the window size and the threshold, you can filter out the random noise and make the real diagonals pop up. So still, reading a dot plot can be very subjective. It does not give you the single best alignment in a mathematical sense. In this situation, dynamic programming methods do come into picture. In the next video, we are going to learn about the dynamic programming method through the Needleman-Wunsch algorithm for global alignment of the sequences. I hope you liked our content. In case you liked our content, then please do not forget to hit the subscribe button, and also please share this video with your friends, students and teachers. I'll be meeting you in the next video very soon. Keep supporting, and thank you so much for watching this video.